So, good afternoon to all of you. We are discussing, if my memory goes right, and if you are able to refresh your memory, we are discussing about the synthetic theory of evolution. In that synthetic theory of evolution, till now we have studied the theory of natural selection, then especially the mutations, both chromosomal as well as the point mutation. Now we are studying in the last class only about the variation in the population. All of us know that variation is a tool for natural selection, and variation generally arises because of the mutation, as we have already seen. Now, we will first think how the variation is helpful, what kind of variation we observe, and what is the root cause for variation. So, both the discrete and quantitative characters of every individual, they contribute to the variation in the population, undoubtedly. So, uh, this discrete characters for a short period it may be in the population and they may disappear, or even they can permanently. But whereas the quantitative characters vary along the continuation of any population. As far as one study is concerned, this quantitative characters contribute more to the variation in a population. These variations are due to genes, the genetic variation. Now the question is how we will be able to measure this variation. What is the frequency of variation in a given population? Actually, the gene variations or genetic variations are generally measured as gene variability or nucleotide variability, because the gene variability refers to the variation with respect to the nucleotide sequences. So that's why it is not wrong if you say it is the nucleotide variability, which we call genetic variation. And in the when I talk about the gene variability, in the sense the average heterozygosity, not homozygosity, but heterozygosity, is the measure of average percent of loci that are homozygous in a heterozygous in a population. What percentage of loci, what is the average percent of loci in any heterozygous population? Very rarely we see homozygous population. Generally, mostly it is the heterozygous population. And what percentage of loci shows the gene variability in a heterozygous population? For this matter, you have to study the nucleotide variability. For that, again, you have to go for in-depth analysis of the DNA sequences of the pairs of individuals. If there's a pair, male and female, you have to study the DNA sequences of both, and then you have to measure the nucleotide variability, because DNA sequence study will give you the differences in especially the nitrogen base arrangement. One can observe variation between the two populations also. What is population? Population is a set of species. set of individuals of a species, of a particular species, in a given definite geographic location as well as in the time frame. When I say two population, they generally exhibit geographic variation. How? To give one example, elephants are found in India, I mean Indian subcontinent, and Africa. They are found in two different geographical regions. And if you study the genes of Indian elephants and the African elephants, you can see nowadays the variation between these two populations. Even externally also, phenotypically also, most species exhibit the geographic variation. Many times by looking into the external feature of an organism, you can say that this must be from this geographic region. Geographic region. Why it is so? Because they come from different gene pools. African elephants have their own gene pool. Indian elephants have their own gene. As a result of that, there's a difference in the gene pool. Just recollect what we have studied in the Galapagos Island. Each island has its own gene pool. By seeing the beak, you can attribute that bird finches to a specific island because morphologically that is distinct. The morphological distinction is because of sharing the different gene pools. One reason that is Medaria is the home of several isolated population of the mice. And if you study this, each population, one can see even the chromosomal variation among the population also. And this chromosomal variation among the different population of the mice in that region is because of genetic drift, not natural selection. This variation is not brought by natural selection but as a result of the genetic drift. Even some geographical variations do occur as a climb. This climb is a graded change in the trait along the geographic axis. For example, one particular fish, that is mimichok fish, same species, but vary in the cold region and the temperate region. Whereas here the variation is because of the natural selection, but not in case of the mice, it's a genetic drift. Because here the nature has differences. In one it is very cold and in another condition it is a temperate region. So as a result, the fish will show the differences with respect to their appearance, which is because of the natural selection. Now coming to the central dogma, that is the source of genetic variation. What is the source of genetic variation? How this genetic variation arises? All of you know, new genes may not be genes, even the alleles can arise by mutation because we have already studied the mutation or as a result of gene duplication also. ಓಕೆ ಕೇಳ್ತಾ ಇದೆಯಾ? ಜಸ್ಟ್ ಟೆಲ್ ಮಿ. ಕೇಳ್ತಾ ಇದೆಯಲ್ಲ?
ಹೌದು ಸರ್.
ಓಕೆ.
ಹೌದು ಸರ್.
Yes, sir.
The only mutation in the germinal cell can pass on to their offspring because germinal cells are responsible for the production of male and female gametes. Suppose if the same mutation happens in the somatic cells, however good or bad that mutation may be, that is restricted to that individual only and will not be able to pass this character to the future generation because all of you know the somatic cells will not take part in the reproduction, sexual reproduction. A point mutation is a change in one base in a gene. One nucleotide base changes means it brings the point mutation. Then what will happen? The effect of the point mutation may change. It can vary. Suppose if the mutation happens in a non-coding region of a DNA, absolutely it has no value. Most of the time, often no harm, often harmless. That such mutations will occur and it has no relevance to us also. Because the change in the nitrogen base sequence or the mutation in the nitrogen base sequence has happened in non-coding region of the DNA. Many times the mutation in the gene can be neutral because the redundancy in the genetic code. You know that there are same code, same genetic code for more than one, for one amino acid, you have more number of genetic codes. So if this code fails, some other code will generate the same amino acid. So that's why if even if it happens, most of the time the mutations will be neutral because of the redundancy in the genetic code. Yeah, but sometimes the mutation which results in the change in the protein production, they are often harmful. Most of the time, I should say that these mutations, suppose if they are able to change the type of the protein that they produce, because proteins are the building blocks, which all of you know, they are often harmful only. And when such harmful mutations take place, generally they are not carried forward. But sometimes a change in the protein production can be beneficial also. But rarely I should agree to the fact. That results in the change in a protein production rarely becomes beneficial. And this may be carried forward. Even altering the gene number or position, which all of you know at the gross level when the chromosomal mutation takes place, either delete, addition, disruption, rearrange, which may, which many loci are typically harmful. Even this. Changing in the rearrangement of the many loci on the chromosome. Most of the time they are typically harmful. But sometimes there's a duplication of small piece of DNA. If a small piece of DNA gets duplicated, what happens is it increases the genome size. The increase in the genome size is usually less harmful. I don't say that it is not harmful, it is less harmful compared to the rearrangement of many loci. And when they are less harmful, they may be retained. And when they are retained, a further mutation may happen to that, these duplicated genes. When they are exposed to further mutations, any new mutations, it may take up a new function. It may take up a new function. And it may be preferred also. Just to give an example, to detect the odor of anything, we have genes. This gene has duplicated number of times. The duplication has taken place in humans less and mice more. In human, we have more than almost 1,000 copies of the gene, duplicated genes. In mice, it is 1,300. Now, another fact I would like to draw your attention to the other fact also. Mutation rates are low in animals and plants. One mutation in every 1 lakh genes per generation. Remember, it's a very low because there is a proofreading mechanism during the protein synthesis. Even if there is a mutation happens, it will not allow that to happen. There is one mutation every 1 lakh genes per generation. Even mutation rates are very lower in prokaryotes, but in viruses they are very high. So that's why the mutation rate is in virus is very high. So that's why we are failing in finding vaccination for many of the diseases because they are mutation. Virus show the mutation, as you have seen in case of the AIDS virus. Till now, we are unable to bring any concrete vaccination because once you develop the vaccination, by the time you develop the vaccination, the virus mutates because rate of mutation is very high in case of the viruses. Then these mutations, if they have to pass on to the next generation, it is through sexual reproduction. Because not in the sexual reproduction, there's a reshuffling of the existing alleles to new combination. We have already studied the recombination of genes. There's a reshuffling, even swapping existing alleles into new combinations will happen. In fact, the organisms which are reproducing sexually, recombination of the allele is more important than the mutation because recombination is bound to bring variation because this is going to take part in the sexual reproduction. But when the gene mutation takes place, most of the time it may be redundant or it may be harmful or it need not be in the germ cell, it can be in the somatic cells also. But whereas the organism which reproduce sexually, there's a recombination during the crossing over process. That becomes more important to us than mutation, and the genetic differences it produces make more adaptation possible in the hospital. So sexual reproduction has more role compared to the mutation. Now the question is how to test whether an evolution is occurring in a population or this Hardy-Weinberg equation is generally used to test whether the population is in a static state or a fluid state. That means whether the population is evolving or not. Where the evolution should happen, it should happen in a population. First, that's why you can apply this Hardy-Wimber equation to a population. Then you can clarify whether this is happening or not. Then what is population? Population is a localized group of individuals of the same species, capable of interbreeding and producing fertile offsprings. Population means it should be the members of the same species, where the members should be able to interbreed. Not only interbreed, they should be able to produce fertile offspring. That means the offspring should also be able to produce the offspring. That is, we call it as a population. A population has its own gene pool. Sum total of all the alleles of all loci in a population, they forms the gene pool. Then the total number of genes and the alleles on all the loci in a population, we call it as gene pool. Suppose if the individuals in a population are homozygous, loci is fixed for the same allele. It is at a particular region. Same allele, locus is fixed on the same for the same allele. But it is not so in a heterozygous population. But in homozygous population, it is yes. The locus is fixed if all individuals are population for a same allele in a homozygous population. See, this is a herd range of Bedford seas. This is the antelopes. This is the porcupine area, that is 40-mile area. These are the two populations. See here, this is the porcupine herd, this is the 40-mile herd. They are of the same species, but they are two different populations in two different geographical ranges. The gene pool of this herd is different and the gene pool of this herd is different. That's why you can see the differences because the genes shared by them are different. This is known as the porcupine herd. This is known as 40-mile herd. I hope you can see the differences. Now, in this heterozygous population, in these two populations, the frequency of an allele in a population can be calculated. The frequency, what is the frequency of allele in a population can be calculated. For any diploid organism, total number of alleles at a locus is the total number of individuals times 2, 2 times. Total number. So, the total number of alleles, if you are find out, and the number of individuals multiplied by 2. The total number of, in any allele, there may be dominant allele and there may be recessive allele. The total number of dominant allele at the locus is 2 allele for each homozygous dominant individual plus one allele for heterozygous individual. The same logic applies to recessive allele. Probably it is not very clear to you, I know that. How many total number of dominant alleles you have in a particular locus? For example, if tallness is a dominant allele, how many dominant allele or other character may be dominant? You find out all the dominant alleles at a locus and two alleles on an average for each homozygous dominant individual and one allele for each heterozygous individual. Heterozygous individual naturally will have one dominant allele and one recessive allele. Same applies to the recessive allele also. Homozygous recessive 2 and heterozygous recessive, if it is there, it is 1. By convention, if there are two alleles at a locus, P and Q are the two alleles at a locus, are used to represent their alleles. There are two alleles. The frequency of allele A is P and allele B is Q, if you see. The frequency of all alleles in a population will add up to 1, not more than that. For example, P plus Q becomes 1 here because P and Q are the represent different frequencies, and whatever may be the frequency, P plus Q equal to 1, because frequency of all alleles, if you in a population, if you calculate, it will add up to 1, not more than 1. So that's why P plus Q equal to 1. So now you take one example. Consider a population with wild flowers and take one allele, that is for color. If it is incompletely dominant for color, how it will be? There is, in a population, there will be 350 red flowers, 160 pink flowers, and 20 white flowers. Means red flowers, homozygous, that will be CR and CR. When in a heterozygous condition, it will be pink flowers. In homozygous recessive condition, it will be white flowers. Now the number of copies of each allele, by applying the same formula, how much? Any dominant, here I have told you the total number of dominant alleles at locus 2. Locus is 2 alleles for each homozygous dominant individual plus 1 allele for each heterozygous individual. See, if there are 322, you have to homozygous 2 plus 1 allele, that is for heterozygous condition, it becomes 800. 350, 320 into 2 plus 160 will give you 800. For recessive allele also calculation is same. 20 into 2 plus 1 heterozygous, that is 160. 200. So now CR is 800 and CW is 200. Now you calculate the frequency of each allele. Here, the frequency of CR divided by 1000, because totally it is 1000. 800 plus 200, 1000. 800 was the, what you call, the number of copies in, we have calculated the number of copies of the each allele. So 800 copies divided by 1000. That means 0.8. For the recessive, it is 200 divided by 1000. It becomes zero point 0.2. And I have told you the sum of alleles is always 1. 0.8 plus 0.2, it gives you 1. This Hardy-Wimberg population principle describes a population that is not evolving. Then this is Hardy-Wimberg equation. It should be 1. It is 1. If the population does not meet the criteria of Hardy-Wimberg principle, then you can conclude that the population is evolving. If it meets the criteria or condition for Hardy-Wimberg principle, that is, the total number of alleles will not, is always 1, then that population is not evolving actually. Because now we should know what the Hardy-Wimberg equilibrium states. Principles of alleles and genotype in a population remain constant from generation to generation unless they are forced by external factors. The frequencies of alleles and genotypes in a population, they remain constant from one generation to another generation. They are not going to change unless there are external influences. So you may say that, see here in the earlier this one, this is zero point 0.8, the frequency of dominant, and recessive is zero point 0.2. And after some generation, all will be dominant because these two recessive will be vanished. Because their contribution is only 0.2%. This is 0.8%. When there's a random mating, this will contribute more, this will contribute less. As a result of that, future nth generation may be everything this gene only for the CR, the CW will vanish. But that's not happening because Hardy-Wimberg principle states that frequency of alleles and genotype in a population remain constant from generation unless they are affected by some external pressure. In any given population, when the gametes contribute to the next generation randomly, if it is selective, then it can change. But I am talking about random mating. In natural population, for that matter, I should include the word natural population. The gametes contribute to the next generation randomly. When there's a random contribution, allele frequencies will not change. Mendelian inheritance that preserves genetic variation in a population. How? Just you can take. Now, alleles, the frequency CR allele is zero point 0.8, frequency of CW allele is zero point 0.2. The alleles in the population. What is the chance? An egg will have 80% chance of CR allele and 20% chance of CW allele. Even the sperm will have 80% chance of CR allele and 20% chance of the CW allele. If you bring the random mating, again that comes to 88.8 and zero point 0.2. So, constant frequency of alleles in such a gene pool. For example, the same population of 500 wildflowers and 1000 alleles where CR equal to zero point 0.8 and CW equal to zero point 0.2, the frequency of genotype you can calculate. That is algebraic formula. What is that? P square plus 2PQ plus Q square. CR means 8 square 0.64. CR CW, that's the heterozygous condition, 2PQ, 2 into 0.8 into 0.2, it gives you 0.32. In homozygous recessive condition, Q square, zero point 0.2 square, 0.004. The frequency of genotype, you can confirm it by using the Punnett square. See, here it is 8%, this is 20%. When the sperms with CR will have 80% and sperm CW they will have 20%. When you put this in a Punnett square, 80% that is 64 and 16 heterozygous condition 4%. What will have? In the first generation 80% CR zero point 0.8 and the 60% zero point 0.2. And what will have? 64% homozygous dominant, 32 heterozygous and 4% will be homozygous. See 80%, 80%, 16%, 4%. So 96% this, 96%, 4% this. The heterozygous CW condition. Again you can see here 16%, 64% plus 16% CR. It will give you 80% because 50% is CR is the heterozygous condition, where 50% is the CR allele, 50% is the CW. So 80%, and in 4% homozygous condition, but 16% is heterozygous CW, 20%. That is 0.2 P. That means what we have assumed, because of the dominant this thing 80% is there already. So in the future generation, this 20% is not going to be selected. No, for generation to generation, the frequency of alleles remains changed unless there is an external pressure. If P and Q represent the relative frequencies of the only two possible alleles in a population at a particular locus, it will become P square plus 2PQ plus Q square equal to 1. Where P square and Q square represents the frequencies of homozygous genotype and 2PQ the frequency of heterozygous genotype. Now, whether we will be able to test the Hardy-Weinberg equation. So in the earlier this thing, the population is not evolving because P square plus 2PQ plus Q square equal to 1. There is no net change. In the first generation also there were 80% CR genes and 20% CW, and that has continued from generation to generation. It is not going to change. Now, the first step in testing whether evolution is occurring in a population or not. Sorry, there is some. Now, one back. There are certain conditions. Hardy-Weinberg theorem describes a hypothetical population that is not evolving. It is in a hypothetical condition what we have discussed. In real population, alleles and genotype frequencies do change over a time. What we have taken was the hypothetical condition. Now, we will take up the real population where allele and genotype frequencies they change over time. I have made it very clear. Hardy-Weinberg population equilibrium can operate if there is no external pressure. So, what are the conditions for a non-evolving population? There should not be no mutation. There must be always random mating, not selective mating. No natural selection, and the population size should be large enough, extremely large population, and there should not be any gene flow from other gene. These are the conditions. If these conditions are there, so that's why I have told you there is no external pressure. If these conditions are not met, population will show evolution. In a non-evolving population, this is more theoretical. No mutation, no random mating, sorry, always random mating, no natural selection, exceptionally large size population, and no gene flow. If all these things are there, population will not evolve. But in nature, it is not possible. As a result, natural populations can evolve at some loci. In some loci, they may evolve, while in some loci, they may maintain the Hardy equilibrium, Hardy-Weinberg equilibrium, where there is no evolution. Some loci evolution, in some loci there is no evolution. So uh, we can assume one example, but the time is not permitting me to uh, take that example. I will continue that in the next class. So now let me know whether you have understood what I have told you, because it involves
Yes,
some
sir.
sort of mathematics. Only thing, this ratio of 0.8 and 0.2 will continue from generation to generation. But what we think, this is 0.8 and this is 0.2, when there's a random mating and all those things happen, next population will have from 0.8 to 0.9, and ultimately it will have every population of one type, CR only, not CW. But it's not happening. That's what I wanted to. And as a result, the population will be not showing an evolution because there is no external forces. It follows or meets all the conditions which I have told you. There is no mutation, there is always random mating, there is a large size population, there is no gene flow. All these things, when these things are there, it is true. But in nature, it is not happening, and I will demonstrate that again with the mathematical example in the next class. Do you have any doubt?
No, sir.
Okay, then shall we meet in the next?